This protocol is used for mapping and discrimination of the primary and process transcripts in maize mitochondria. This technique includes an RNA normalization step, and it minimizes the influence of unstable 5' triphosphate that hinders a clear discrimination between the primary and process transcripts.
Besides maize mitochondrion, these methods could be applied to plastids and other plant mitochondria.
Start by collecting 20 grams of developing kernels into a 50 milliliter tube on ice. Transfer the kernels to a pre-cooled mortar and add 10 to 20 milliliters of ice-cold extraction buffer. Grind the kernels completely, add more extraction buffer, and filter the ground tissue through 2 layers of filter cloth. Centrifuge the filtrate at 8,000 times G for 10 minutes, then transfer the supernatant to a new tube and discard the filtrate. Centrifuge the tube at 20,000 times G for 10 minutes, pour off the supernatant, and resuspend the pellet in 6 milliliters of wash buffer. Aliquot the suspension into 5 1.5 milliliter RNase-free tubes, and centrifuge them at 14,000 times G for 5 minutes. Discard the supernatant and freeze the mitochondrial pellet in liquid nitrogen. Extract mitochondrial RNA using commercial reagents according to the manufacturer's instructions, and set up an RNA 5' polyphosphatase treatment. Then, recover the RNA.